We're very happy that Ilya is here. We are continuing on from where we started earlier this summer. We had a 10-week-long summer bioinformatics course, which was very well received. And that was also a collaboration between LBRN, CCT, and Biotech. We had the summer course sponsored, the seats were sponsored by the LBRN, about 25 seats at that time, and another 20 seats by CCT. So based on our feedback, we've expanded this to now potentially a 6-month-long course, which will culminate in April sometime, coinciding with our annual Bioinformatics conference. And this is an orientation seminar, which will give you an idea of what you can expect over the next 6 months, what these different modules will be including, and you will have a week starting tomorrow based on this talk to go ahead and register for this course. Once again, this time LBRN is sponsoring 50 site licenses for primarily undergrad institutions, the LBRN Network institutions, and the CCT will sponsor 50 spots for people around LSU, people at LSU, and please take time to put the registration in and give a reason for why you think this would be useful for you, and then we will make an assessment on the top 50 applications. And without further ado, Ilya.
Thank you. Yeah, so first of all, I'm really happy that we get to continue this, because during the summer we only covered a limited number of topics, and this time what we want to do in the webinar is just go through exactly what the program will cover, how it's structured, and then hopefully we'll get to talk a little bit about what this will produce by the time of the conference, which is, I think, an exciting opportunity as well. So the first thing that I want to point out is that this is an attempt to kind of bring together several things that LBRN is already trying to do, right? So this is not going to be a traditional course, like, here's a textbook and we will cover several topics. What we will try to do is work through projects. So, and the other aspect of it is that we're really trying to make it work online as well as offline for all the different participants across the state. So the program that we are trying to design as a result of this experience is something that would work for different levels of students, faculty, different levels of students, people without any background in bioinformatics and people with background in bioinformatics, are really trying to bridge the gaps between those different types of people. And the second thing I want to mention is that this is a model that we're trying at a few different places, so not just here, hopefully gathering experience from those different locations and making some sense of that. A core piece of this program is this bioinformatics platform. I'll show you a little bit more about it later, but the idea behind the platform is really to provide a single interface for people that are not familiar with coding, that don't really know how to work with data or databases and things like that. Also not limiting you to whatever laptop computational power you have, so it's really a centralized place. And I'll expand on that a little bit more on how we use that to also track progress and help people at different timelines. So the program right now, this platform has several different types of accounts. The people participating in this program will have an educational account that essentially limits your ability to upload large data sets, but we will provide uploaded data sets for you to use and practice during the course. Now, over here on this side, you see the different areas of analysis, so essentially different data types, and they reflect some of the topics that we will cover during the course. So the idea is that you know, again, going back to that idea of projects, is that you have an educational data set that is provided to you alongside a publication. You understand the biology, you know, how the data was generated, why it was generated, all of those things from the publication. Then we discuss an approach, a possible approach to analyze this data. So process the data, prepare it for analysis, analyze it, get some results. And then really we have an ability to just know what we're expecting, so we can check whether what you got is what we were supposed to get. But also we can do a discussion around the results. What do they really mean? You know, why are we looking at them, etc. And then you have an ability to, you know, kind of talk about what some alternative methods could be and why would you choose other methods instead of this one in your analysis. So that process is followed by an online curriculum, right? So this is just one of the lessons, Transcript Topics 1. What you see here on 1 is just an outline, so a more traditional outline of topics that we are covering. And then inside you have, you know, how is this data generated? What does it really represent? You have different terms that people typically are not aware of, so we cover all of those. There's a glossary. And then we kind of map out exactly what are we trying to do and what are the different scenarios that we will be discussing. And you always have assistance on the site. So the idea is to really take this functional platform, take the online courses, and kind of bridge it all together. Now, how exactly are we doing that? So, for example, if we take a topic, so RNA-seq, for example, there is some text, a video introduction, you understand basic concepts, and then you have several example projects that you can choose from. Those projects are already on the server, so you don't actually have to go and download and upload anything, so that we just give you the reference links. And then you have a number of pipelines that you run to analyze the data. Not very clear here, but basically you have processed output, so you work with tables, you visualize them, and then you have tests and comparison with what other people are doing and with what we know should be happening of the analysis results. These are, again, different types of projects from publications that have either a specific data type, or some of them actually have multiple data types in a single project. So here are some examples of data sets that we will be using during the program. One of them right there is a publication. We started talking about it in the summer course. It's called Modeling Precision Treatment of Breast Cancer. Really, the idea is how can you use RNA-seq, exome-seq, proteomics, how can you use those data sets to determine whether a patient will respond or not respond to a treatment, and what is a better combination of data sets to predict effective response. The data is all, again, like I said, uploaded on the server, but different types of data sets are available. So you go to RNA-seq, to transcriptomics, you analyze one portion of it, you go to genomics or exome-seq, you analyze that data, and then in the analysis section, we talk about different methods that could help you combine those data sets together and understand how they could be used in treatment. Now, obviously, this is a big topic, right? You need to understand oncology, you need to understand different breast cancer subtypes in this example, and you need to understand the treatments and what do they actually do, and why do we need to, why is there a challenge to predict response? So all of those things are discussed on the modules, either under the project or in the transcriptomics modules that I'll talk a little bit in a little while. And then you can see that other data sets as alternatives to that one really talk about similar ideas, right? So you have right there, analysis of the change in immune response in cancer. So this is really a study of macrophages and the role of macrophages in the development of cancer, how the immune system can either support the development of cancer or suppress it. You have a PDX model study where you really look at the mouse models where a lot of these tumors are studied. And then you also can look at a data set from TCGA, the Cancer Atlas, the Cancer Genome Atlas, where actual patient data could be looked at. So the idea is to provide these data sets at various levels of complexity. You start from cell lines, you go to animal models, and then you look at it in patients. You also look at it from a standpoint of different data types. So you look at RNA-seq, exome-seq, proteomics, and then how to integrate this data and how to apply it in the real world. And you also understand the context. So in the context of oncology and the challenges around precision medicine, the challenges of precise diagnostics and treatment selection, how do you actually use this in the real world? So this is the idea of this project-based course. Now, this is kind of an example. This is a lot like what we did during the summer, but now expand this to a number of different topics. So just let's take one of the topics as an example, transcriptomics. For each topic, we will have an orientation workshop, kind of like this, talking about, what is transcriptomics, what are we studying, etc., just basic terms so that everybody's on the same page. Then you have several weeks to go through the online courses, and all of that culminates in a workshop that is a hands-on workshop where we actually take data sets, we analyze the data sets, we compare results, and we talk about the challenges that arise. Then you have some time to work through your own project, right? So all of these modules provide you with some practical exercises. Try this, try that, kind of different examples. At the end, the idea of the independent project is to really go through all the stages on your own. How do you do them? How do you plan your analysis? Why do you make a decision here like that in the context of this project? And why in the context of another project your decision should change, right? So we hope that this format allows you to learn some basic terms, and then try to not be limited by a template. Just because I know differential gene expression, I don't just apply it in every single problem that comes up. And how do I try to generalize those concepts and start thinking creatively with the methods that I'm familiar with? And then after the end of that independent project, we have a workshop. Again, in some cases, we will invite guest speakers that are experts in that specific domain. For example, in the metagenomics course, Chris Taylor from LSU Health Sciences will be joining and talking about his research and the things that he does. And so we hope that this also provides you with an opportunity to evaluate. Before this, let's say I didn't know a lot about metagenomics. I went through the course. And now when I hear an expert talk about this data, I can really understand and appreciate the types of things that he talks about and the type of research that he does. So now the idea is to do several things in parallel, right? So we understand that some of you maybe participated in the summer workshops, some of you didn't. We will try to give a few options. So like Ramesh said, it will be around a 6 months course. During that time, we will cover a number of topics, but they will kind of overlap. So you'll be able to choose, I want to work on this project or on that project. And if you already completed the coursework, you can actually just take the projects themselves. Also, through the independent projects, we typically work with those groups. Instead of doing a whole group of 100 people, we'll do smaller groups and discuss specific challenges within that project group related to that data set. So what are the topics that we will actually cover? I hope that the text is clear here, but I'll go one by one what these topics are. So again, like I said, this is the areas of analysis of the platform, but it's also a good way to view what are the topics that we'll cover. So the top over there is transcriptomics, RNA-seq and RNA-seq2. Basically, it's the same idea, just some alignment methods have been added that are not that are database related. Underneath, you have RNA editing. So the idea that you have gene expression, isoform expression, that would be under transcriptomics. Then you have several events that happen, afterwards, after the transcription. And so that would fall under RNA editing. We also will talk about single-cell transcriptomics. So we'll try and use a data set on single-cell gene expression and isoform expression. Then over here you have ChIP-seq. So it's the chromatin immunoprecipitation data. Really, how does the DNA, how is it folded, and how does that affect the transcription of genes and isoforms? We will talk a little bit about bisulfite-seq, but not a lot. Primarily, we'll focus on ChIP-seq. And then we will also have a whole course on genomics. So right here, these two are mutation variant. So your SNPs and somatic mutations is what we will focus on. But also copy number variations. So CNV, copy number variation, multiclonality. So how does the the same gene, if it's encoded a number of times in different places in the DNA, how do you analyze that and what do you do with that data? We'll also talk about microRNA and long non-coding RNA, so RNAs that are actually not genes or isoforms that don't make proteins, and we'll have a whole course on metagenomics or the microbiome. Now, if you know Chris Taylor and the course that he published, it will be kind of based on the same topics, but it will be a lot more practical, and you don't have to do any coding to actually do the practical exercises. And we will expand a little bit to metagenomics itself, so not just the 16S, but also expression of these different elements. Under genomics, we will also cover touch on, maybe not cover in depth, but genome-wide association studies, GWAS. And we will talk about what do you do with these tables, right? So a lot of these methods are actually processing the raw data, right? So it's helpful. You have to understand how to process the data, but then you have to also know what to do with this data, right? You have these huge tables, somebody generated for you. What do you do with them? How do you get to a result that is interpretable? So we have over here a number of data mining tools. They will be methods like dimensionality reduction, PCA, TSME, things that help you really visualize data as you process it. And also it will be things like clustering and network analysis, things like that. Also on the supervised analysis, we will talk about classification. How do you take this data, and if you can annotate it, how do you use that to classify new data that comes in? So really, an introduction to machine learning, you'll learn some concepts that are kind of standardly used for these types of data sets. And also what are the limitations of these methods, and how do you verify that the method produces something that you can annotate, and to look at it from the biology side of things and understand whether it has relevance or need. So a little bit more in detail, I want to stress the point that on one hand, this is intended for people that don't want to learn Linux and how to install new things on your laptop and really skip that whole part and try and work with this interface that allows you to quickly compare multiple different methods. So just to explain what I mean by that. In every case, so right, there's probably thousands of methods out there. And all of these methods have a little bit of a variation of the same idea, right? So for example, you can do alignment in many different ways, or you can do differential gene expression in different ways, or you can annotate in different ways, right? So how do you know which method to select? So two things about that. First of all, like I said in the beginning, we will be providing with some standard tools, right? So like you have a FASTView file, do this. And then once you do that and you see that for this project it worked well, but for this project it didn't do well, why is that, right? So then the question becomes, what alternative methods do I have, and how can I logically substitute one method with another, and how can I compare and evaluate the outcome? So that's what this tool is really built for. You can see that everything is organized into sections. So mapping on genome has a number of alternative mapping on junctions. You have things that do both things. So they're really trying to develop a logic of why these tools are placed in the same category. And every time you select a method, it also gives you that explanation. So you can go back to the paper if you're motivated. You can read through the analysis approach and really try and understand why or why not. And also, you'll notice that there are multiple ways to connect the things together. Not everything is possible. And so that is where we try to eliminate those typical mistakes when people are trying to force two things to work together that are just not going to work together. And so it hopefully cleans up some of those things. But except for transcriptomics, here are a couple of examples for genomics. So you have mutation variant on this side and copy number variation on this side. Again, the same idea, you have to map your file, but then you have a million ways to call the actual systems, right? So which one do you use? Maybe you can select a bunch, just like right there. You have 3 different methods to call variants, and then you create a VCF file. And in the VCF file, you will get the statistics for the mutations that were identified, and you can also compare the results. The same thing for copy number variation. So this is genomics. Here are a couple of examples. So you have metagenomics pipeline, and you have the ChIP-seq pipeline. So what you see in these examples is really that the interface is going to be the same. It's going to be a standardized way of doing things. But at the end, you will always be able to go back, and a lot of these methods, you can identify them. So if you want to create now your own pipeline, you can go and do it in bioconductor or any other environment that you're more familiar with. Now, an important part of this, as I mentioned before, is going to be how do you work with the table at the end, and how do you share your results? How do you visualize your data, either to understand it yourself or to present it to somebody else because you want to communicate the results of your analysis of your project. So we'll talk about, things like creating a heat map or generating a dendrogram. How do I interpret these things? What is the height of these different lines that connect the samples in this example? Why are they separated into these trees? And what does that mean? How do we interpret that? This is an example of a PCA. How do we understand these dimensionality reduction techniques, and what are they actually telling us? How can we use these methods to understand not just the relationship between, for example, samples, but also why are they separated, right? So kind of feature selection using these methods. So some of these things we already covered in the summer course, but we are going to do a complete review of what we did in the summer for transcriptomics, but also we'll have an expanded course called machine learning, and that will be just about the method. So no matter what kind of data you feed these models, something will come out. How do you evaluate what comes out and whether it's useful, but also what methods are more appropriate for what types of data. So really trying to understand the data types, how do they differ, and what methods make more sense. We'll also talk about building pipelines of machine learning methods, right? And I think that we're doing a course with Georgetown interacting with those students is really helping us define the areas that I think are important to address. So the idea is that you have, again, machine learning methods, you feed them something, something comes out all the time, right? Whatever you give it, it will produce, just like differential gene expression. If you want to find a difference, some difference, ultimately you will be able to find it. So how do you know whether it's giving you something relevant that you can actually rely upon? So we'll talk about how to combine these different methods together to evaluate and answer those questions and find something more reliable. So if you're thinking of biomarker discovery or gene signature construction, those types of ideas, not just related to, whether I do it with 10 samples or 10,000 samples, that's not the only limitation. It's also how to use the methods properly. So again, just to talk about the structure of this program and the workshops and the types of interaction that we think is effective. So online learning has a lot of limitations, right? You go online, it's kind of on your own schedule. Most of the time it's when you're tired and you want to go to sleep, or you want to go to eat, or you have other things to do. So there's a lot of interruptions. And at the same time, not doing the online things, what we've learned from doing these workshops here as well, is that people that don't take the time to do the online stuff really slow down everybody else. Right? They come in with very basic questions, they don't understand terminology, we cannot all move at the same time. So two things about that. One, we did surveys, 80% of the people think that even if the courses online are more difficult to get through, without them, the workshops have a much less value. On the other hand, we understand that not everybody moves at the same pace and not everybody's at the same level. So what we did is we organized different level workshops and meetings at different locations. You see at Pennington and Tulane, here at the CCT, to really try and work with groups that are a little bit at the same level. And also online, right? So not just limited on location, but because I know that LBRN institutions are spread out across the state, we are not limited to just doing the meetings here, and also we can do those online. And these are just some examples of what we did. Another point that we wanted to stress is that bringing together information about how people interact with the data and with the lessons is really helping us adjust the pace and address specific challenges that people face as they go through the projects. So just so that it's clear to everybody, this is the back end. Everybody's activity is being analyzed, and we use it to address any challenges, but also to help individuals that want to eventually do some actual research, right? So things go wrong, why do they go wrong? Or people that just can't get to the next level. So we try and go through that. And over here, what you can see is we create a community that will all go through the same things, and you can actually track your progress against other people's progress. We track things like enrollment in a course, enrollment in a project, completion of the project, passing your test, all of those things, as well as running pipelines. What we've seen in the past is that people that do the practical side of things really move fast with the content. People that don't end up really having a difficult time translating that knowledge to projects that they take on and require a lot of assistance. Now, I don't know everybody's level here, but typically what happens, I think, in the biology space is that you get your data, and you know that there's a bioinformatics core or some person that's good with computers, and you kind of rely on them, here, do this for me, right? And you typically get a result that you don't exactly understand, right? So you kind of look at the table and you think, okay, this is nice, what do I do with this, right? So a lot of it is because of that communication gap, right? So you speak the language of biology, and that person speaks the language of computer science, or the other way around, and how do you communicate? And I think that, one of the things that we're trying to develop here is a way to communicate more efficiently, Because in the end, even though you will know some of these methods, does not make you an expert bioinformatician. You still know some ideas and some methods, but maybe just enough to make that communication a lot more meaningful for you and for that expert. And so, or maybe the other way around, maybe you are the expert, right, and the biologist, and you don't really know how to explain to the biologist why this method and why not that method, right? And so we hope that eventually, after this course, some of you will continue to do independent research, utilizing the infrastructure that I think has been built up through LBRN and through the LSU CCT, taking these things further into getting actual results for your publications, grants, whatever else you might need. At the same time, this course is not going to be an opportunity to just work on your projects, right? So we did have a number of people that just came with their own data sets and said, this is what I want to do, and kind of ignored the rest of it. So I know that that's ultimately what most of us want to do, is work on projects, but unfortunately this course is not going to be that opportunity, but we will be able to focus on that later down the road. The other benefit, I think, of this course is learning to read through the papers without skipping the methods part, and really understanding what they mean when they show a figure or when they mention a specific method or a long list of methods that they use to analyze the data. What we did with pre and post assessments is we basically took those same project publications that we will be using as examples, extracted from there both the conceptual side of things, so why were they doing this research and what did they achieve with this research, and the technical side, what methods did they use, why, what were the considerations around data standardization, normalization, etc. So all of those concepts, and just measure the results from the people that we surveyed, hoping that at the end of the day, you going through the courses and working through the data sets, you are able to understand what a figure like that means and why it's significant and how to interpret and evaluate it. Right? What are the limitations of this research paper? Why is it important? And how would I do my own research in a similar way? What's exciting about this course specifically for me is that a year ago, just over a year ago, we started working on this program. We kind of in 2017, around September, we started officially transitioning into this specific area of education. And I'm happy today that we are implementing this program in a number of universities, as well as working with over 2,500 students worldwide. I think this is this is a poster that we presented at the previous LBRN conference, where we just did a pilot at the Loyola University of New Orleans. And today, I think the exciting part, at least for us, is that starting this course with a bigger number of students and really, again, trying to prepare for that conference is an exciting opportunity to really see what we've achieved. That's for us. But I think also it's an opportunity for people that want to participate in this course. Being aligned with the conference and being aligned with other activities that are happening through LBRN and through LSU Center for Computational Technology, I think is an exciting opportunity to showcase what you can do or what you want to do, and also what is it that you're finding, right? So the idea is that maybe by April, when all of this takes place, we can have project ideas at least. We can have an understanding of how to collect the data for that next project and how to plan for that analysis, or where can you find some online resources that will help you at least validate some of these ideas that you've got. And also then it will help us establish a way to start addressing those challenges. One thing that I want to say about this first pilot at Loyola that was very interesting is that we realized that for a lot of faculty, a major limitation for them to get the most out of their research data are the students. And what we found was the students that showed a lot of progress during the course then easily found mentors and collaborators to start working on that research data. So if you're faculty, bring your students here. Once they know what to do, you'll be able to help them use those skills on your research data. If you're a student, you want to do a summer course with a mentor and actually, put your name in a publication, that's the opportunity to do it. You go back and you tell them, this is the project that I did. This is how you can get involved in that kind of thing. And so I think that ultimately, those are the types of results that we expect, especially in a community like this that brings together faculty, students, and students of different levels and different backgrounds, right? So we have students from finance background, from computer science background, so really technical things that have no relevance to biology. And we have bench lab, PhD students that really just wanted to do more with their own data. So that's an exciting thing about that. Now there's going to be a week-long process for the registration after this event. And what I think we decided is that we will post a schedule of the different data types and different programs that we will announce. But just about the registration itself, maybe I can ask John to talk about how do people register and who do they talk to about their registration.
So we're going to have a sign-in sheet for everyone who's attending. I've sent out the link for the sign-in so we can have you all opt in and give us your email addresses. Obviously, we'll publish a registration link. I apologize. I'm listening to the delayed stream, so by the time you said John, a couple of times, that was really weird. But I need the course information to send that. So when I have that from you, I can do that.
Right. So tonight or after this meeting, we are going to be talking with Gus, Dr. Kuslos, and Ramesh about the dates and how to align those courses. But pretty much what we said is there's going to be an introduction to bioinformatics, which is going to be very broad and general. And if you know something about bioinformatics, that's not a course that you have to do. Might be interesting for you. It really talks about how data has transformed life sciences and technology in general. So,
but just following up, yeah. So I have a sign-in link that, and if I finally got the QR code, you can scan and just sign in after this so that we get your contact information for those online. Please check in the chat on ViewMe/live for that link. But obviously we'll be emailing that sign-in opt-in link out so we can get your information so we can go and just send that.
Do you want to say a few things about registration before I transition to the other part?
Yes. I'm sorry, I just stepped out for a moment because I thought somebody called about this and was not able to connect. So I'm sure Elia and John have identified that registration which begins tomorrow. Another thing is all these talks are also recorded for you, for those of you who are
yeah, we hope to have that available by tomorrow or the next day.
Yeah, and so when people sign up, please make sure you write another little note about why you want to participate. That will help us assess. And try to get in your applications at the earliest so that we have an assessment of how many people are interested. I don't know if we have PUIs also online, right, John? Or we have the LBRN PUIs also online right now?
Well, we have, I mean, it's whatever names they put in.
Okay.
Eduardo is watching the stream. So
great.
some people are watching live stream. We have people, various connections.
Okay, great.
I can't tell you exactly who's doing it.
Okay, great. So LBRN PUIs too, please encourage your students and faculty members to participate. It's a great opportunity. And go ahead.
Okay. So if you want to register today, Dr. John, if you want to register online, put your email down or some contact information, we'll send you that soon. So before I kind of go on to show you a few of the things that we talked about, does anybody have any specific questions or comments so far?
I think that all sounds really, really great. Having done some of this stuff without this program, it's very, very time consuming. The program, is it going to be, how would we have access to that after the program if we become trained in how to use it?
It's a question to Ramesh, I think, but the general idea is that we already have educational accounts that will be transferred to like a research account after, I guess, the 6-month program. But it also depends on whether you're part of LSU or one of the PUIs. So all the technical details, Ramesh.
So one of the ideas behind this is a lot of what their platform uses is open source software. So we'd like to believe that at the end of this, you will have some idea of how to use other open source software out there. And then we are also looking to get into collaborations with Fine Biotech on making this platform available. That will come at a later stage of our collaborations in future. So right now we are concentrating on the educational module. There's another parallel track of interactions. We are looking at research and research support and stuff like that. Does that answer it appropriately? That they will be able to, these are open source software that you're using, and they will have the ability to use.
And I'll show you just in a moment how to use that and how to utilize that learning to later on do your own project.
Great. Thanks.
Anybody else? Any questions before I go? Any comments? Okay. All right, so just to demonstrate to you a little bit practically so that it's all clear, this is the educational platform. You can already go there. It's edu.t-bio.info. You can go right here under courses and preview some of the courses. A lot of them are freely available. So what makes this program special and different from the freely available courses is that you will actually be able to do the practical stuff, right? There's just the videos and the text and things like that. And the other thing is that we structure it around these workshops, right? So I think it's very important that you do the online, you practice by yourself, you end up with a lot of questions, basically. But then it's good to come to the workshop and be able to go through some of those things. You will notice that some of the things here come with a price, but as a part of this program, all of that will be free. So you'll just have a code and none of, you don't have to pay anything. So that's going to be here. You'll see that we're working on a number of specializations so that the projects and the concepts align into some meaningful direction that, if you're a plant biologist as opposed to oncologist, those are a little bit different. So, aligning the data and the ideas with that will be here. You'll see the events, the different workshops that are coming up, those will be posted here. And then, of course, here are some examples of the projects that we'll be using. These are just examples. So during the course, we will actually do workshop presentations of projects, and then you'll be able to get the data sets. Down here, you will also see that you will have a community. And once you are registered through John, you will be able to view the curriculum here, and you'll have a forum, and you'll see everybody's progress. So some of the things that I showed before, that will be right here, and you'll be able to interact both with us, asking questions or telling us about something that's not working as expected, and also between yourselves. And it's important to note here that the courses are not a textbook. They're not static content. A lot of times, a lot of people have a question on some specific topic. We create a video or a lesson about that, but we post it here under your organization. And okay, so do you have any questions about this? Basically, if you are a part of LSU or LBRN, you have to use your account, your email associated with the university, so that the accounts are synchronized and you know what it is. If you just want to take a look around and use your social media account or something like that to log in, you can do that as well. But once the program starts and you're registered, make sure it's the same, because we had a number of confused participants last time. And the second thing is the platform. So I'll just demonstrate a couple of things so that you know what to expect. All of you will, again, get an account. So you'll be able to see the data that you have and the pipelines that you've run and revisit that for the duration of the course. Again, an educational account has limitations as to the size of the data that you upload. So primarily you'll be dealing with tables. You can upload any table, but in terms of raw data, like a FASTQ file or something like that, you'll only be using already uploaded data on the server. So as an example, we'll just take RNA-seq and I'll show you how the system works. You will have this area on top always to define the data type that you're going to be working with. So for example, I want to use with human data and paired reads in FASTQ format, then I'll be able to, for you as an educational account, add file is not going to be working. So what you'll use is URL bulk upload, and then you will have these SVL files, which are really links to a path on the server where the files are stored. So what you'll see is, in reality, all of these FASTQ files are already uploaded there. And now I can, if I want to do differential gene expression, I can drag them in groups. I'm not going to do that right now. And now you can start building your pipeline. So to build your pipeline, of course, you need to start with pressing start. And here you can read a little bit about RNA-seq. But then you see that some buttons are darker, some are lighter. So the darker ones are the ones that you can select. They will actually work in your pipeline and for the data that you uploaded. And so, if I'm doing RNA-seq, I'm probably going to want to clean up the raw data and remove some of the adapters, remove the...
So those, if you go back one step, you see those darker boxes and the lighter boxes?
Yeah.
So is that determined by what you have uploaded by the software? So that would happen even if you are at a later stage when you're using the platform for your own data. Then the software will analyze your data and accordingly make whatever suggestions based on these data boxes. Okay.
Yeah, and you will see that every step of the way it changes. Mm-hmm. So it knows the data and it knows what output will come from what you've already selected, so you won't be able to make the most obvious mistakes. Yeah, so for example, if I want to pre-process my data, and here, I use Trimmomatic. If I want to use Trimmomatic in my research and I saw that was effective, you can go down here and most likely you can download that. I mean, not most likely you can. Then I would want to clean up my PCR, and now that I've done that, for example, I would want to just map this on transcripts. So once I select bowtie to T, you can see that my options are very different, right? So I'm not going to go back and do simulations, for example, or go back to top hat. That doesn't make any sense. And then I'd be able to quantify my expression in a specific number format. And we'll talk about all of those things in the course, but you can also see that all of these are organized into sections, and you can read about the section and exactly what the section does, so that conceptually you understand why one thing goes after the other and what exactly are we trying to do. And after you're done, you can click on Save. I will name this. And then I can run the pipeline. So once I run the pipeline, I can go to My Pipelines, and you can see that this one is prepared. So I can always go back and see exactly what my selection was, and also what my input files were. So if I go back to another example here, I can click on Uploaded Data, and I can see what I uploaded in this pipeline and what was I trying to do. You can also see what was kind of the setup right here, right? So you won't see it for those that are just prepared, just for the ones that are already completed. And then I will get a PDF report that tells you how long the jobs took and what exactly they did, and some figures if those are available, and you can go directly to the downloaded or to the download section to download the results. Now, another question that a lot of time comes up is, how do I use this if I, for example, know how to code in R, or if I'm a little bit more of a coder, don't just want to look at the files myself. And so what we've done in some sections that are relevant is we actually provide you with the source code right here of whatever was done. So you download the zip file, and then you can just open this in RStudio. And what it will do is it will actually walk you through exactly what happened in the pipeline, right? So if you can just read, I mean, I think any of us, just looking at this, you will see with the comments exactly what was happening, how did the data get loaded, and what were the inputs. So the same inputs that I had on the platform, how are they registered right here? And you will notice that in the download, you actually have the source file as well, and you have, so source file, and you have the call. So if you want to call it from the terminal, you can do it right there. Right, so the idea is that this platform is not going to lock you in forever. Once you learn how to navigate the different sections, you will be able to decide what we want to do on some infrastructural level using a centralized system like here, or if you want to take it further into your own data system. Now, the other thing that we will do is we will talk about, so for those of you that do not want to mess with R or anything like that, we will utilize Excel a lot, and we will try and use Excel's functionality like t-test or summary statistics and basic things that you can do in Excel to really understand how can we just rely on the platform and tools that most of you will likely have to really be effective with the data, right? So we'll do things like filtering, we'll do things like, evaluating different statistical measurements on your data set, and we'll talk about visualization, how to create basic visualizations in Excel, and how to prepare them for something like a publication. So that will be another part of some of these lessons. Again, going back to the areas of analysis before we finalize here. So what I mentioned to you, and so like I said, you will have an account. That account will be associated with the group. LBRN will have probably multiple universities or LSU. And then for at least 6 months, the duration of this course, you'll be able to use this primarily on tables and small data sets. The sections, again, that we will cover, and we'll send out the actual schedule and we'll post it on the website, right? Something like this week.
Yes.
today or tomorrow. But you will have, in parallel, Introduction to Bioinformatics and RNA-seq. We will have RNA editing and epigenetics, again in parallel. And then we'll have metagenomics and genomics. And so those are going to be the core courses, as well as the separate course on machine learning. So the separate course on machine learning will be available because the methods will be utilized throughout these different sections. It will be available for the whole time, and we'll just refer to it during the different sessions that we have. So just a review. So like I said, RNA-seq. We will do RNA editing. We will do a little bit of bisulfite methylation, but primarily ChIP-seq. And then we will do mutation variant, which is under genomics, as well as copy number variation and genome-wide association. We will touch on that as well during the genomics course. And then we will look at microRNAs and metagenomics. So those will be the courses under next generation sequencing. And then how to analyze that data later on. We will have some supervised methods like classification, unsupervised methods like clustering and dimensionality reduction. And you will also see under utilities that we have different things like normalization, visualization, gene set enrichment analysis. So just useful scripts that could be used during the process. It's important to say that for those of you that actually program or have experience with programming or specific scripts that you think are a must-have for a specific purpose or a section, this system allows you to incorporate those scripts and make them as one of the buttons very easily. So if you do have something like that, talk to me and we'll try and make that happen. Just to demonstrate to you, these are all the different patterns. So you can see here, image analysis, for example, consists of 6 different scripts, right? So those 6 different scripts, I can add them, modify them, etc. And essentially, this becomes easy to use with the same input system that you've seen up in the phone. So I think that's it on my part. We have covered the topics or the data types that we will do. We covered how those will be delivered. Again, it will be an online and offline with workshops as kind of the connecting point. And it will be focused on projects. So our hope is that as you go through the projects, you'll come up with your own ideas for projects. And then the next phase, we'll be trying to put together students and people that you could work with or you could work by yourself and eventually do some great research. So that's it for me. If you have any questions or comments.
So you saw the penultimate slide that Elia showed, which had a listing of the courses, some of them going on simultaneously. We'll have that available on alongside our registration link. And go through it. We'll also have a link for the talk. Perhaps if you want to refer back to it for some of the details. And then the registration link officially goes live tomorrow. So if you have any questions, feel free to contact Elia. Elia's email ID is there, me, and shoot us your questions and we'll try to answer them as best we can.
I will understand today. So definitely here is some participants actually from the research about the plants. So as your sample data is animals. So do you think it's taken to some of the animals?
Plant, yeah, we do have examples of plants. So we have a project on potatoes and Arabidopsis. So some examples that will be somewhat relevant, obviously not exactly what you do.
Any questions from the remote sites?
I guess nobody's
good.
All right, I see something happening there, I don't know why. But if there are no questions, then you can send your questions by email. We hear you clearly. Any questions?
No, I think that's it.
All right. So if you have any other questions, you can shoot an email and we'll try to answer as best we can. Thank you all for attending. Thanks, Alim. William, don't forget. Yeah, William, thank you.
So uncomfortable with that. I think
That sounds, because